Today we'll first introduce Vibrio. Well, we talked a little bit about Vibrio on our first lecture. We talked about the story of Dr. Zhengzhou. That's the time before people even knew bacteria. And there is some pandemic of Vibrio cholera in London, and people were trying to figure out what caused the pandemic. And at that time, people thought that it's the dirty air or something smelly in the air caused the disease. But Dr. Zhengzhou used a map and also statistic methods and figured out that the disease is due to something in water, in a well. And later we figured out that cholera is a bacterial infection. So that's the story about Vibrio. And today we are learning the pathogen Vibrio, and we'll learn how they cause disease, their pathogenesis, and also how we can prevent cholera. So Vibrio is actually the name of the genus. It includes a lot of species, various species. This is the common structure of Vibrio in this genus. And you can see they are bacillus, but they are not straight. You can see this comma-shaped bacillus. And for the gram stain, they show this red color. So Vibrio is a gram-negative bacteria. And there are a lot of species in Vibrio genus. But we'll focus on Vibrio cholerae. Vibrio cholerae is the pathogen of cholera. And that, while cholera was once one of the major threats to human life. And we'll focus on Vibrio cholerae. Okay, here is some general knowledge of Vibrio cholerae. They are gram-negative, we mentioned, showing the pink or red color. They are comma-shaped or curved rod, like this. And they are highly motile by means of single polar flagellum. You know, we learned flagella a lot, but flagella is, the word flagella means a lot of flagellum. And here they are polar flagellum, means that by the end of the bacteria, there is only one flagellum. And this will propel the bacteria to swim in medium, in liquid. And when you check the bacterium under microscope, you can see that they can swim fast. It's motile. And when you culture the bacteria on agar, on the solid medium, they form convex, smooth, round colonies like this. And they grow well on TCBS agar. TCBS agar is a special medium designed to distinguish cholerae, Vibrio cholerae. And the agar itself shows a greenish color, but when Vibrio cholerae grow on it, it shows this yellow color, golden yellow color. And that golden material can diffuse into the agar. So this is a typical medium. And TCBS is short for thiosulfate, citrate, bile, sucrose. That means in this agar, it contains these 4 different material. Vibrio cholerae can ferment sucrose and mannose. That's the 2 different types of carbohydrates. And they are oxidase positive, meaning that they can produce oxidase. They grow, or they prefer, a very high pH, around 8.5 to 9.5. So That means that cholerae is alkaline philic. They prefer the high pH above 7. So it's alkaline philic. And they don't like acid, and acid can kill the bacteria rapidly. This is interesting because Vibrio cholerae is transmitted through through ingestion. But you know that in our stomach, we have a lot of acid in it, and the pH is very low, around 2.5 or something like that. That means a lot of cholerae bacteria you ingested into the stomach will be killed. They cannot survive. They will be cleared out. So that means only a large number of bacteria can cause infection, can set up infection in our human. And also we know that those bacteria are also halotolerant. They can tolerate a lot of or high level of sodium or sodium chloride. So it's halotolerant. For their antigenic structures, so that's the components on the bacteria which are antigens, which can stimulate the immune system to respond to the bacteria. One is the H antigen. That's not surprising because they are motile and they have flagellum on each bacterium. So the flagellum carry proteins, and those proteins are antigenic. But those proteins are heat labile, meaning that they cannot, they are not resistant to high temperature, heat labile. So that's the H antigen. And another one is O antigen. For Vibrio cholerae, O antigen is very important because it's O antigen. The O antigen is from the specific polysaccharide on the very outer layer of LPS, and that confers a serologic specificity. And there are so many various types of O antigen. Right now, we have identified more than 150 O antigen serogroups. More than 150 O antigen serogroups. And within these over 150 serogroups, only O1 and O139 serogroups can cause disease in human. And those two serogroups, we call them classic cholerae. Classic cholerae. And also, O1 antigen can further divide it into 3 sub-serotypes. I think this is based on their location where identified. Except for the classic cholerae, there is another biotype called Altor biotype. And Altor is another pathogen, another Vibrio cholerae pathogen, which can also cause disease in human. Altor biotype produces hemolysin, but it is resistant to polymyxin B. Polymyxin B is a type of antibiotic. Okay. And the disease or pathogenesis it causes is, well, here it's oral route. We mentioned that they can only infect humans through the oral route. And because they are afraid of acid, and we introduced that only a large number of Vibrio cholerae can cause infection in a person with normal gastric acid. So in our stomach, most of the bacteria will be killed. Those bacteria do not reach our bloodstream, but remain within the intestinal tract. In our lumen intestinal tract, they stay there, but they can produce a toxin called Vibrio toxin, and Vibrio toxin can cause disease, and the illicit of the disease is cholera. And later we'll introduce the cholera toxin and how it causes disease in human. Cholera toxin, it's an exotoxin, means that it's a protein secreted by the pathogen. It's heat labile enterotoxin. And again, this toxin is an AB toxin, meaning that there are two types of subunits. One is subunit B, which binds to the receptor of the host cell. And here, for the B subunit of cholera toxin, it is GM1 receptor. It's a receptor on the cell membrane. And also subunit A. Subunit A is the effector subunit, which can cause toxicity in the human cell. So those are two different subunits. But interestingly, for cholera toxin, there are 5 identical subunit B and 2 subunit A. And 5 subunit B increases the opportunity that it can bind to the receptor on the human cell. Okay? And once the B subunit binds to the receptor, then the A subunit is translocated into the cell. And in the cell, it can function by transferring or catalyzing ATP molecule into CAMP. That's a cyclic AMP. And we learned on our cell biology class that CAMP is one important molecule which can regulate the behavior of the cell. And here in the epithelial cell in our intestinal tract, the increased CAMP will lead the cell to secrete large amount of water, large amount of sodium and potassium into the lumen. That will increase the level, the amount of water in the lumen, and that causes the diarrhea. And this is only the first step. When the water in the cell is low, more water will diffuse from the blood into the epithelial cell. So you are losing the water not only from the epithelial cell, but from all over the body, from the whole system. So that's why cholera is so lethal. And if not treated well, it can kill the person within a short time. So this is cholera toxin. And clinical findings, the incubation period for cholera is varies in different patients from 12 hours, so that's half a day, to 4 days. But one interesting and important property of the disease is that the onset of the disease is very quick. So it's sudden onset. So the disease the symptoms shows out quickly with nausea and vomiting and profuse diarrhea with abdominal cramps. So that means it's very painful in the abdomen. So this is the first property. Another property is that the diarrhea it caused looks like rice water stool. So that's rice water stool. If you analyze the diarrhea, it contains a lot of material. Not only large number of Vibrio, it also contains the mucus. So that's the intestinal mucus, which means that it damages the mucus and also the peeled off epithelial cells. So it will cause damage in the lumen. So that is why it's so painful. And it can also cause dehydration of the whole body and also circulatory collapse. And the patient doesn't have any urine because the patient loses so much water through diarrhea. This is clinical findings. And when we test the existence of Vibrio, we can run some laboratory tests in the lab, and the specimens collected usually it's stools. So that's easy to understand. And once the stools is transferred into the lab, we can first check the morphology of the bacteria with a microscope, and you can check whether it's motile, and you can stain the bacteria and see whether it's a coma-shaped bacterium. So that's a quick examine. And we can also further culture and purify the bacteria by culturing it on agar, on a solid medium, including blood agar with a high pH, because they prefer a high pH. Or we can use a TCBS agar, and they can produce yellow substances on that agar. And also, we can test the patient's serum, or we can test the bacterial antigen. We can use known anti-O1 or anti-O139 anti-serum to and mix them with the bacteria. And if the bacteria cluster together, and we say that that's the agglutination of the bacteria. That means that you can identify the specific serogroups of the pathogen. For the epidemiology of and prevention of Vibrio, we should learn some history here. Cholera, six pandemics of cholera happened between 1819 to 1923. So that's around 100 years. There are 6 pandemics, and no wonder during those 6 pandemics, it killed a lot of people. And for these 6 pandemics, cholerae 01 is the pathogen, the classic biotype. But there was another pandemic happened in 1960s. It began in 1961. So we call that 7th pandemic. And this one, the serotype of the bacteria is biotype Altor. So that's a different one. The Vibrio cholerae biotype Altor. During the cholera, the infected persons in particular eliminate large numbers of pathogens. Even when the patient recovered, when the patient is recovering, the convalescents may also shed bacteria for weeks or even months after the infection has abated. So that is why during the cholera pandemic, there are so many bacteria all over the environment, and people need to be very careful to prevent cholera infection. Transmission of the disease is usually via food, and in particular in drinking waters. So we need to keep our drinking water clean, and we should improve the sanitation, particularly of food and water. And this is usually it's a problem in developing countries or undeveloped countries, because in those countries the sanitation condition is not good enough, and sometimes people doesn't like to drink the boiled water, and either due to lack of fuel or that it's a lifestyle, they just drink the raw water. And that is a major problem, because the raw water might be contaminated by Vibrio cholerae. And once there is some patient infected, we should isolate that person, and so that they won't spread the germ everywhere. And also their waste or excreta must be disinfected. So we stop the the transmission from one person to the environment and to other person. For cholera treatment, the most important procedure to take is give the patient enough water and electrolytes. Cholera is so dangerous because of dehydration and loss of electrolytes. So we should replace the loss of those material. And that is a very important and essential supporting treatment to help the patient to recover. And meanwhile, we can also give the patient antibiotics to help them and to kill the bacteria, including the antibiotics include the tetracycline and doxycycline. Those are the first lines of antibiotics people can use. And keep in mind that the most important procedure is to give your patient supporting treatment. Give them enough water and electrolyte. Okay, so generally we covered the topic of Vibrio cholera. And with the left time, I would like to show you a very short cartoon. It's a short video. Shows us if a cholera happens in a village, in some area, how people can prevent the disease and how to cure the infected patients. And we do have other materials, other videos to watch, but we don't have enough time to play it here. You can log into the Superstar platform and watch other videos. Okay, let's see this story of cholera.
This is the story of how cholera changed my village.
Can you hear the audio?
Tiny germs of cholera, too small to see, spread through the river. So small, yet so dangerous. Without realizing, women carried cholera home in the water. Flies carried cholera on their feet. Unwashed hands spread it too. We swallowed cholera germs in our water, on our food, and on our fingers. It happened so fast. By morning, my father was very sick. He had diarrhea that looked like gray water and poured out of him. I was so scared. I went for help. I never rode so fast. One look at my father and the nurse knew it was cholera. We had to work fast to save him. We made a special drink to help him. First, we made the water safe. We filtered it through cloth and boiled it for 1 minute. Then we mixed half a teaspoon of salt and 6 teaspoons of sugar in 1 liter of this safe water. It tasted like tears, not too salty. I worried my father would die before my eyes, but he soon felt a little stronger. The nurse explained to me that not everyone who swallows cholera germs gets sick like my father, but they can still spread the disease. Now I needed to take safe water to my village and teach them how to protect themselves from cholera. I saw a girl carrying water. I told her she could make the water safe by adding chlorine drops and waiting half an hour. There was a man about to eat with unclean hands. I told him to always wash his hands with soap and safe water after going to the toilet. Only with clean hands could he eat safely. I saw villagers spreading cholera into our river. I told them we needed to dig latrines far from the river, at least 30 meters away. This was important to keep our village clean. I found a mother preparing unsafe food. I told her, first, we must wash our hands with safe water. Then, we had to wash and peel the food, cook it, and always eat it hot, and protect it from flies. I spread the word throughout my village and ran to find my father. I was so happy to see he was better. Our village became healthy. Now we filter and boil our water to make sure it is safe. We always use latrines and always wash our hands after. Food is safe from flies, washed and peeled and cooked. And we always wash our hands before cooking and eating. We made our village safe from cholera. Spread the word. Your village can be safe too.
Okay, so that's the story of cholera. And in this series of videos, I think we should have another video. That's the story of Ebola. And I think I have uploaded that video on the Superstar platform. And hopefully you will have a chance to watch that video. Okay, so basically that's all for this topic on Vibrio cholerae.